My name is Max. I'm an analyst at LDV Capital, and I'm here to introduce our next speaker, Akhila Kosaraju, the CEO and president of Phare. Akhila will discuss how AI-powered drug discovery is transforming antibiotic development. Her keynote is titled Supercharging Drug Discovery and Advancing Critical New Classes of Antibiotics with AI.
Thanks so much, Max. It's great to be here, and I'm very excited to share what might be a little bit of a diversion from our current conversations to really talk about one of the oldest problems in medicine and how we're supercharging drug discovery and advancing critical new class of antibiotics exclusively with AI. So Phare was launched in 2020 off the backs of an extraordinary discovery out of Jim Collins' lab at MIT, where he discovered the first novel class of antibiotics against the superbug, so the most deadly and drug-resistant bacteria in the world, using artificial intelligence. And this really electrified a field that has been in a valley for a number of years, both from a commercialization perspective and a innovation perspective on the R&D side. We've been at this for a while now. So, with 5 years of experience and sort of predating this current wave of AI innovation, we feel that with AI and antibiotics, there are a number of lessons learned across the healthcare and life sciences field. So just to level set everyone, there's $7 billion in VC investments in 2024. That represents a 3x increase from 2023. 95% of pharma companies report investing in AI capabilities. And yet this is still very much a nascent, although it's exploding, it's a nascent field with 0 approved AI-generated drugs to date. That's not an indication of failure in any way. It's just an indication that we're really at that hockey stick inflection point of the tremendous progress in the field where you'll see exponential growth and I think reductions in time, cost, and greater efficiency in drug discovery and development. But I want to focus on one key takeaway for this group, which is that I think the most important assessment of how well AI can really accelerate a particular life sciences process or technology is the fit with the problem that you're trying to solve. And that was really the light bulb moment for us back in 2020, that AI and antibiotics are an exceptionally good pairing of a problem and a solution. So specifically with AI, our basic methodology is we train our models on compound structures against particular bacteria and train the models on 0 to 1s on how well a particular compound kills a particular bacteria. And we also are able to train our models on identifying novel chemical structures. So train the model on what an existing antibiotic looks like, but ensure that the model doesn't just predict for identical chemical structures or scaffolds. And so built into the model is novelty. So novelty of prediction in predictive AI or novelty of design if we're actually using generative AI to design our own compounds. And novelty is the core of the issue with antibiotic resistance. So you may have heard, about this looming crisis in antibiotic drug resistance. There's predicted to be 39 million deaths by 2050. It will be the leading cause of death in the world, matching cancer. And the issue is that we're using the same old antibiotics with the same old mechanisms of action. And clearly our bacteria have been smart enough to figure out mechanisms to evade all these, existing classes. So to be able to train our models on novelty is a huge win. And again, gets at the core of the challenge in this space. We also have a rapid feedback loop where we can validate our models in a petri dish. So again, pairing a frontier technology with one of the oldest challenges in medicine, using minimum inhibitory concentrations, one of the oldest visual technologies in the life sciences, where you examine a petri dish under a microscope and see how much of the bacteria a particular compound is able to kill. We can then feed back the results from that model into our AI platform quickly and efficiently. And I think it's incredibly well suited, again, for rapid iterations. And I think part of the reason why we've been able to be a first mover in this space and have a lot of great progress. And then the last piece I want to mention is that given that it's such a low profitability field, we can de-risk our drug candidates earlier in the process with computational capabilities, which are much cheaper than all of the assays and the, significantly costly preclinical process that can cost up to $10 million, considering all the failures along the way. So we want to take fewer shots on goal, better shots on goal, do them faster, and actively feed that back into our model. So the next thing I want to mention is that every part of our process, and I think this also applies to any life sciences company that's incorporating AI, is pairing human and machine intelligence. You will not get to a point anytime in the next 10, 20 years where you don't need that brilliant scientific expertise from the beginning to the end of your process. So again, we conduct wet lab work to test and curate our data. We feed that data into our computational models. And then coming out of the models, we again use our medicinal chemists and our microbiologists assess and prioritize amongst the molecules that are being generated from our AI platform. So and throughout every phase of this process, there's visual tech in examining the Petri dishes, in assessing the compound structures, and in again retesting the compounds to ensure that they are not the equivalent of hallucinations out of a model, but actually synthesizable and developable drugs. And the last thing I want to mention on the technology side is that we've moved from predictive to generative AI. And you likely have heard a lot of talk across life sciences about this boom in generative AI design compounds. And again, we're in the early days. But for us in particular, the ability to not just predict previously unknown antibiotic properties across broad compound libraries, but to actually design our compounds from scratch has again enabled more novelty, getting to the heart of the antibiotic resistance problem. But it also enables us to incorporate translational filters. And I just want to make a finer point on this. So what that means in a nutshell is that we are moving from targeting bacteria in a targeted strike that's very good at killing bacteria, but it's a drug that you would never introduce to patients because of how toxic it is, to actually incorporating those toxicology and drug-like parameters into the training of our models. And from a human perspective, this brings the patient to the beginning of the process. And I think at every step of the way for every life sciences company, to ensure that the models and the technology that you're using are solving a human problem, I think is really the best way to assess what will win this race to get the best drugs ultimately to patients in the fastest time frame, in the way that really serves the greatest unmet need. And then, given that we can now design our own compounds using generative AI, we're no longer constrained by compound libraries that have been picked through by virtually every company and academic group out there. Public libraries and libraries that exist in academia. Now that we can actually design our own compounds based on our trained models, again, we have orders of magnitude more potential novelty, and we're not inhibited by the prior art of the IP in many of those libraries. And the last thing I want to mention that I think AI and antibiotics is also a first mover in, and I feel that we are really trying to lead the charge on this, is sparking a revolution in open sourcing drug discovery data. And I think that the pharmaceutical space is a little bit far behind and siloed in terms of treating all of our data as proprietary. And when we started Phare Bio, we went out to a number of big pharmas, and as you can imagine, they just don't want to share their compound libraries because that's gold. That's really what much of their pipelines are built off of. And our proposition is that you can actually share a number of the parameters and the toxicology, the pharmacokinetic, the solubility data, the formulation data. Much of that is commoditized data that could be helpful across drug discovery for any therapeutic area. So at Phare Bio, we are generating hundreds of thousands of data points that we will open access, and also open access our generative AI models. And we're hoping to spark a revolution in how drug discovery is done, really trying to lift all ships, because we think that, like other areas of tech, we can all move faster together if there is more of a culture and a process around data sharing that wouldn't compromise IP, but really leads the way for again, getting drugs to patients faster, which is ultimately our goal here. And as we're looking at the fundamentals of the healthcare system with antibiotics, procedures like C-sections and treating a child with strep, basic surgeries, cancer, chemotherapy, all of these medical technologies that we really take for granted are built off of the proposition that we have good antibiotics. So we're here to solve a humanity-altering challenge. And we feel that AI will really help us get there faster, more precisely, and with better drugs that actually address the core of the problem with antibiotic resistance. My prediction in 20 years, as we develop this technology for this particular challenge, is that we will ultimately have these open access data sets that can be broadly applicable, sharing these commoditized data points, and that the drugs that come out of our AI models will be ever closer to ultimately what goes into patients, that we'll be effectively printing these drugs that are usable and really can change patients' lives.